Okay, novel native variation to breeding pools. Okay, long titles. Okay, so the work I'm presenting here, I'm largely going to focus on maize, which is the crop I work on. This project is called Seeds of Discovery, which has been largely funded by the Mexican Ministry of Agriculture, SAGAPA, and involves a wide range of partners. From CIMMYT, we've got Dave Marshall from James Hutton, Andre Killian from DART, Ed Buckler from Cornell, and John Hickey from the Roslin Institute. Nope, doesn't work that way. All right, we're not moving forward here, guys. There we are. Okay, so essentially, we've already heard about a lot of the issues facing food production and human nutrition. Now, when we consider a lot of the germplasm banks that we have, we see that a lot of valuable genetic variation is untapped. I mean, it remains on the shelves of the gene banks. The gene banks across the CG system, the international systems, and national gene banks do a very good job of disseminating large numbers of accessions and varieties out, but largely the requests coming in are for elite lines that are housed in banks or germplasm that's already well characterized and well described. So gene banks are at the moment largely museums, but they shouldn't be museums. Genetic variation is the basis of all breeding. Without genetic variation for a particular trait, you can't really do breeding. It doesn't make sense. So what the Seeds of Discovery project looks at is really leveraging genetic resources and feeding that into crop improvement programs. Okay, now, new genetic resources aren't going to solve the issue. There's a lot of work along basic crop improvement, agronomy and seed systems work that also needs to go along hand in hand, and there are other projects that look at this. So SEED looks to systematically identify and mobilize the beneficial genetic variation into breeding programs to accelerate and strengthen genetic. What I'm going to describe today are three main modules that we work on. There's the molecular atlas, which looks at really describing a lot of the genetic variation that we have, identifying underused accessions that may be of potential benefit, identification of new alleles and allele donors, and then this, the key component, is really the development of bridging germplasm. A lot of the materials present in gene banks are very wild and wonderful, and certainly in the case of maize, very few breeders are willing to really engage and start to use the land races there. There's too much linkage drag that comes along with these things. So we need to repackage the germplasm into a more breeder-friendly component. Alongside the three main components, there's a big component on information management. There was a question in the earlier session, what do you do with all of this data? How do you manage it? And then capacity strengthening. It's very well for us to develop these tools to use within the CG system, but we need to ensure that a lot of our partners in the national system are just as easily able to start to use the data and generate benefits from it. So, the molecular atlas. I think of gene banks as supermarkets. Okay, they should be a place where you can go, you know what product you want to go in, you can go in, look at the variety of products there that match your criteria and make a selection. At the moment though, the gene banks largely look like this. You can see tins on the shelf, but you don't know whether you're going to go in and get a tin of beans or a tin of soup. Yeah, there's no real way of accessing and understanding what you're selecting from the gene bank, which is why a lot of these materials remain unused and unrequested. What we're trying to do with the molecular atlas is this, give some basic information to enable people to make a better selection from the gene bank. And that is being done using a common label of genotypic data. It will be lovely to be able to phenotype the entire gene banks, but it is just out of scope in terms of costs. Yeah, we can't genotype or phenotype everything for every characteristic we may be interested in. We've got to find a way of rationalizing the collection first. So with the genotyping, we've been using GBS as the main platform, and it's configurable. Obviously with GBS you've got to try and get that balance of the number of markers you're interested in and the number of samples. Yeah? So we've been using two different flavors of GBS. We've been using the variant that Cornell University have been using. They use APK1 as the main enzyme for the library construction. And with this we get lots and lots of markers. So around 850,000 markers per sample, which is great. Now in this sampling, because they do not have a great depth of sequencing, we can only look at SNP markers. You can't really look at presence-absence variation. And there's a lot of missing data, around 50% missing data. So often in these cases we use imputation. And in this case we've been applying this to genomic selection practices and to GWAS applications. Now when you're studying diversity, especially in the case of an outbreeding species like maize, we want to be able to score heterozygotes. We want to be able to look at presence-absence variation. So we worked with DART and the DART-seq platform, and we've developed new technologies or new flavors of GBS where we have obviously fewer markers, but with those markers we have much higher coverage, lower error rates, less missing data, and unlike the variant we use at Cornell, it's not dependent on B73 as a reference. Around 40% of the tags containing SNPs that we generate have no alignment with B73. And by throwing that information out, as we do with the Cornell system, we're certainly inferring a lot of ascertainment bias. Now, thinking about things like GWAS, we would like some alignment. That would be very helpful for us to actually know where a lot of these are. So in a parallel project, we've been doing some resequencing from accessions and double haploids generated from accessions with Longbio, an institute based in Mexico, and then we're working currently with NRGene on alignments of that material. So for the maize diversity surveys, the big question we had was, can we use DNA pools? Okay, we've got a largely heterozygous crop. There is more genetic variation within an accession of maize than between accessions. So it would be very helpful for us to look at DNA pools rather than have to sample a number of individuals. We had a pool-based method that Marilyn Warburton and Alleles Chakosay had developed from SSR-based analysis. So we wanted to see how far we could take that forward and apply GWAS to the same system. We took a number of diverse accessions, 8 diverse accessions, and we genotyped a minimum of 92 individuals per accession. And then we made simulated sampling of this from 4 to 40 individuals and looked at a number of distance and diversity indices. So here we've got representations of just 2 of the accessions. Here's Cephalophorza-Edwards distance, and as you can see in both cases, as you increase the number of accession or number of individuals sampled from 4 to 40, you see a decrease in the basically standard error that you get around the estimate of distance. Even with 4 accessions, you get a reasonably good estimate of genetic distance. We just tidy that up a little bit by increasing the numbers. However, when you look at a lot of the diversity measures, and here we have heterozygosity, really we still don't saturate or get a representative population measure of heterozygosity with 40 individuals. Okay, if we look at the mean here from the 92 individuals, with 4 we really are underrepresenting the heterozygosity. So what we'd like to do is really use bulks and see how far we can drive this technology to get better estimates of populations. We then went on to actually start looking at realbox, and we generated composite samples. So this is not a composite of DNA, this is an actual leaf bulk that is then extracted in one go. So with the same 8 accessions, we looked at bulks again ranging from 4 to 48. And really here you can see this is a measure of the deviation between 2 samples. So we generated independent bulks comprised of different individuals. And really here from 20 upwards, you don't see any improvement in deviation. Okay, there's about 5% variation that you get between the bulks, and it's not improved by increasing the numbers of individuals. We also looked again at a diversity component, so heterozygosity again, and we used linear and quadratic regression here. So this indicates here the number of individuals at which essentially we reach a plateau. Okay, above that we don't get any variation in the heterozygosity measure. And this was variable. Okay, for some accessions you only needed 12 individuals represented really to get a plateau. Some accessions we never actually got a plateau even at 48. Okay, so there's variation in there. So we take this information and we look at what is feasible in the lab. Yeah, it may be great to put 100 individuals in a composite sample. None of my technicians are particularly willing to punch that many things, nor do we have leaf punches that are small enough. So we came out with an estimate of 30 individuals. Okay, it satisfied the criteria, or was above the criteria required in most samples, and the others, okay, we know there is some underrepresentation and we can live with that. So the composite method that we have gave us very robust measures of genetic distance, well above the thresholds required, and very good estimates of diversity. And essentially what you could see here, comparing the pools with the individual samples, we get a fairly good correlation between allele frequency estimates. This is much improved as we start moving towards higher coverage. So as we can get the actual numbers of sequence tags sequenced in each reaction back, we can actually filter these markers by coverage. So at the moment we use markers that are 5x and higher and have very, very good correlations between the frequencies found in individuals and the corresponding bulk. Now what is the benefit for us with this? We get a 30% fold reduction in cost. Okay, so it's much cheaper for us to start to genotype these materials. And in addition to SNP markers, we can look at presence-absence variation. And these are very, very important for us in order to differentiate between some of the accessions. So what have we done to date? We've now conducted GWAS on the entire CIMMYT maize gene bank. So there's just over 27,500 accessions that have been genotyped. We've done a preliminary analysis of 21,000 of those accessions. We've called in that panel 1.2 million SNPs. And we get a mean of nearly 1 million SNP loci per accession, with only around 20% missing data, which is very nice for us. When we filter using the 5x coverage, we're working on just over 300,000 loci, and that's what the analyses have been based on. Now, in addition to accessions, we've also been genotyping breeding materials and XPVP lines. It's very helpful for us to know what is already available in terms of genetic diversity within the breeding pools to try and work out what accessions may be the most applicable. This is just a multidimensional scaling that you can see here. The accessions are the blocks here in blue. These are all the CIMMYT maize lines. So these are elite lines available from the gene bank. The big block in red here, these are all of the land races present in the collection. So there's very large swathes of diversity that really aren't represented in these elite materials, as we may expect. We did a very quick analysis looking earlier last year at the first 10,000 accessions that we genotyped, and looking at the 540 CMOs that were included in part of that panel. And if you just look at SNP presence, SNP diversity, only 6% of the SNPs that have been sequenced in the 10,000 are present in the CMOs. There's a lot of genetic diversity that we're just not seeing in the elite lines. Now, a lot of that is complete junk. Yeah, we really don't want it there. Breeders have done a very good job of removing deleterious variation. But in among that are some gold nuggets. Yeah, there's a lot of things within this 94% of variation. There are some things that would be very useful for breeding. And our aim is to try and leverage that and move them forward into pre-breeding materials. We have started to combine a lot of the genotypic data with other data resources. We did a lot of curation of the passport data in the bank, and where we have good geolocation data, we're able to derive a number of climatic variables from the collection sites. Using that information, we looked at the aridity index, so it's basically a measure of precipitation versus evapotranspiration, and pulled out nearly 10,000 land races that come from environments with long-term propensity for drought stress. Okay, these are not irrigated environments. We managed to remove those from the collection. Now, no breeder is able to evaluate 10,000 accessions, 10,000 lines perhaps, 10,000 wild and wonderful accessions that fall over and lodge and do other heinous things in the field they're not going to touch. We took the GWAS data and did a quick genetic analysis looking at clustering. We can find 6 main groups represented within the landrace materials. The 6 groups all come from dry environments. Okay, we'd already done that selection. But when you look specifically within each group, there's quite a lot of variation, and some groups come from much drier environments than others. So here we see what we have here is group 4. This collection is largely from highland materials. Okay, now most breeders don't go back to highland accessions. Yeah, tropical and subtropical perhaps. Highland is a very difficult group to work with, but these are potentially the most interesting in terms of new genes and alleles for drought tolerance and heat tolerance. We've developed some breeder-oriented cores. I mean, core collections have been around much longer than I have. And the classic thing is you take 20% of your collection, you do some stratification, clustering, and you derive from that a panel of 20% of your accessions. Now, that isn't always necessarily very helpful for a breeder. What we've done here is look at the numbers of accessions that a breeder is really able to handle in a nursery. The largest here being 1,000 accessions. Okay, so that's around 10% of the first selection that we made for drought tolerance. 500, 250, 100, and even 50 accessions. So these are nested core collections. So you get representation of these 50 within the panels of greater size. So if you're a big breeder, you've got to spare $300,000 in 2 years to do some evaluations, you can just about handle that. If you're a smaller breeder, you can still look at some of this genetic diversity without blowing your budgets. We've started to use the data to look for selection footprints or selection sweeps. Here we've just got a quick representation of some of the popcorn accessions present within the collection. There's only around 700 markers that you can see here, but we can see clear structuration of some of the races coming through. So we're able to start to use this information to look for selection motifs, to look for markers that will help us identify, for example, specific races, which is very interesting for the gene bank characterizations. Okay, onto the alleles and allele donors. When we initiated the project, there was a large panel of traits that came up, nearly 40 traits that breeders were interested in. Now, 40 traits, we can't possibly look at that many. So we had to restructure, and the main emphasis was on the complex traits, the harder things to do. The genetically simple traits, there are some diseases in there, which we've looked at really as examples of low-hanging fruits and proof of concept work. But most of the work is focused on complex traits. We have very little focus on what we call upstream work. So really where we've identified things and we want to look into mechanistic bases, or we'd like to do some gene-based cloning. That stuff we've left really for other partnerships because it's not the primary focus of the project. We developed a large GWAS panel. This is based on around 4,000 accessions. So it's currently to date the most diverse panel available for maize. We took one plant per accession, and we made a test cross of that, which was phenotype. Now, that single plant is not representative of the accession, but across many accessions we have replication of haplotypes. The materials came from 3 adaptation zones, 3 main zones: lowland tropical, subtropical, and highland tropical adaptations. They were materials with genotype. The individual plant used to make the test cross was genotyped, using both the Cornell GBS method, where we really want that high density of markers to try and accommodate for the high LD decay we see, and we use the DART-seq method to help identify heterozygotes. Okay, imputation in breeding lines where you have structured populations is very, very robust. Imputation in accessions hits about 60% accuracy, so we need to be able to back that up with some real data. The traits that we looked at: heat, drought, low N. A number of biotic stressors, Cercospora, tar spot, which is a disease of high prevalence in Latin America, and then grain quality and nutritional quality characteristics. Now, as with all cases, and Rajiv pointed to this, accurate field-based phenotyping is one of the major bottlenecks. And working with land races is not to be underestimated. My colleague Martha Wilcox bust a gut and really generated a lot of high-quality data that we can use for this work. So over about 35 trials, we took just under 1 million data points across 14 different locations. We used flowering time as a proof of concept. Okay, flowering time is not something we necessarily need markers for breeding work, but it's something that has been studied an awful lot. If we could not find the usual suspects panel of genes within the flowering time analysis, we'd have a hard time finding anything else. So a very good PhD student, Alberto Romero, who we sponsored, who's present in Ed Buckler's lab, has been doing this analysis. He used 500,000 SNPs, so took out a lot of SNPs with high proportions of missing data, used imputation with the fill-in, and he performed GWAS on a per trial basis using the BLUPs. And here we've got presentation of days to silking. We have days to anthesis and ASCII as well. We did find the usual suspects, so markers very close to BGT1 and ZCN8. So we thought, okay, it's proof of concept. We can find the usual suspects. What else can we find in the GWAS analyses? What we did find, which quite surprised us, was a very strong new signal on chromosome 4. And this was the strongest hit that we got anywhere. And this is a structural rearrangement. So we had a discussion earlier, or that was mentioned, that structural rearrangements are very important. Yes, they really are important. This is an inversion. It's been previously reported in Teosinte and Highland maize and was assumed to be involved in adaptation. Now, this trial, for any of you who know CIMMYT, will know that Agua Fria is certainly not a Highland location. So the PhD student was really excited. I found this great Highland inversion in these Highland materials. Not in Agua Fria, he didn't. This is definitely a tropical location. So this inversion is well outside of Highland maize. And when we actually look at the effects of this, the homozygote inversion is responsible for nearly a 2 to 3 to even 4 day difference in flowering time. This is the biggest single effect that's ever been found in flowering. So what have we done with other analyses that are more relevant to us? As I mentioned before, tar spot is a particularly prevalent disease currently in Mexico. Climate modelling suggests it's going to spread out of there, it's going to move further south into Brazil and potentially into Northern America, or USA. Mexico is North America. This is what tar spot looks like on the leaves, causes massive necrosis. Essentially you get no photoassimilate reallocated into the grain during grain fill. This is the data from the GWAS panel. These are the two test crosses, the two accessions, Oaxaca 280 and a Guatemala 153. Very, very clean plants and correspondingly good yield. We did phenotype the accessions per se. In addition, again, very clean. Now your yield is a lot lower because these plants are very tall, they lodge, they fall over, they have other issues. When we looked at the GWAS analysis, we found a number of hits that were very interesting. Many contribute quite large proportions of genetic variation and result in quite a lot of clean leaf area. However, all of these are present at a very low frequency within the accessions. So within the panel, these things are all less than 1% in terms of frequency. Okay, very rare things that we're looking at. When we look at drought, again, here we looked at materials and pulled out a number of hits, as you usually do with the GWAS panel. And then we looked to see where these markers were present in terms of, again, here, looking at the CIMMYT maize lines, looking at leaf materials. We found some interesting things. What is already present in the CMOS, there's very little point of looking at those. We found hits right the way across the genome. These regions here, chromosome 2 and chromosome 3, there's basically no representation of those alleles in the CMOs. Further down the genome, 6, 7, 8, and 9, there's very, very high representation of those alleles within the CMLs. We don't need to look at those in breeding terms. When we're looking at which accessions then to start using in pre-breeding or as a best source for these alleles, it's a lot easier for us to find those parts where we already have combinations of alleles. Yeah, rather than developing 3 or 4 different populations, we develop one population. So it's been very helpful for us to look at the genotyping data and identify those accessions which pyramid the sources. And as you can see, these are already very low in frequency. Yeah, less than 5 accessions we could use as these donors. We've also been doing work, I mean, GWAS panels are great, but they're not by any means the be-all and end-all. One of the very interesting areas that we've moved into is per se evaluations. Per se phenotyping is difficult. Okay, as we said, accessions of populations, they lodge. We're trying to look at this across 3 adaptation zones. It's not particularly easy work, but can be very rewarding. We've been looking at drought, heat, and anthocyanin contents. There's a lot of interest in Mexico in blue maize. For the drought work, again, we used aridity index as a proxy for selection. This was done before we had genotypic data, so essentially it's just a broad panel based on racial characterization and AI. And to date, we've evaluated 700 lowland tropical and subtropical materials, and we've already started forming biparental populations in 100 of the best materials identified. So even if you don't have genotypic data, GIS-based characterization where available is a good complement or a substitute for expensive phenotyping. Okay, when we've looked at a panel of materials that we added that were not selected this way, we really do see good gain in using something like GIS or a FIGS-based approach. Breeding germplasm. When we've looked at the development of germplasm, we really have to balance how quickly the breeder needs that new variation with how complex the trait is. Yeah, so there is not one breeding approach that we've adopted. We've used genomic selection, we've tried looking at DHs. We've been using simulations with John Hickey as a way of assessing the best approaches. What founder germplasm to use? Do you go straight from an accession? In the terms of a GWAS panel, do we go to the test crosses that we had? Do we try and make double haploids? What marker densities do we require? Okay, these things have very large effective population sizes. Applying 10,000 markers may not be that effective. How quickly do we need to do retraining in terms of genomic selection? And how many entries do we need to sample per accession, with it being an open pollinated population? Now, we ran this data with a number of colleagues. We managed to break computers that NASA have used for a lot of their simulations. So you can imagine there's an awful lot of data points that we generate. The simplest way of presenting these are regression trees. So if you want to know more about this, please go and harass John Hickey later. But essentially it helps us break down what are the most significant components. And what we've ended up using is an accession-based approach. Okay, doubled haploids are very effective, but they take time to generate, and this isn't easy in accessions. You can generate haploids very easily and cannot get them to flower to double. Test cross approaches are great, but you end up reconstituting the tester. Okay, so we just regenerate the CML testers again, which is not exactly what we want to do. So within the pre-breeding work now, we have 6 genomic selection populations, which are broad accession-based synthetics, looking at drought, heat, and low N. We've got 100 BC1S1s backcrossed to a favourable CML that will be going into phenotyping again this year, selecting 50 of the best materials to go forward and using that data as another basis for genomic selection within these populations. The 2 best accessions for tar spot, we've generated some backcrosses and informing BC1s. And again, these will be re-evaluated genotypically and phenotypically for marker-assisted backcrossing. We can use the initial data that we had from the GWAS panel. We've identified some great alleles there, but there's potentially other variation that we haven't been able to capture because it's just too low frequency within the GWAS panel. And we've ID'd accessions for Cercospora, Tersicum, provitamin A, protein, oil, and fusarium. Now, as I mentioned before, doubled haploid works, but we need to really get these things in a more inducible background before we can really take that technology and deploy it well. Prospects. We want to be able to swim in the data, not drown in the data. Up until now, we've kind of been drowning in the data. Data management issues have been hitting us all. We started to release data. All of this data is available after 2 years of generation. So it's available through a Germinate portal that we're running based on the James Hutton technologies, and through Dataverse. So all the materials available through IP restrictions. We're making available protocols, software and scripts so people can actually start to analyze this data, and I know I've been running out of time, so I'll be quick. We've got a lot of analysis running at the moment, and people are very encouraged to come and speak to us to be part of this analysis. There is more analysis than we have time to work with. And the most important thing really is moving this germplasm forward. So we've got a lot of pre-bred germplasm. The first stuff will be released in 2016, and it's released not only as germplasm but with associated marker information and GS models if they're available. So we are hopefully unlocking the box of genetic diversity and moving those materials forward for people to use in their breeding programs. Thank you.
Well, thank you, Sarah. That was a very interesting talk on how to tap these immense germplasm resources that we have in all of our crops. So thank you. That was very good. I think maybe we can take just 1 or 2 questions. We're running a little behind, but I think we'll take maybe 2 questions.
Japanese have identified early morning flowering trait in wild rice. So did you find such type of trait in teosinte? Because why means in drought and heat conditions, if daytime they flower, more sterility will be there.
With the teosinte, we've got no phenotypic data that we've been using to date. We've got genotypic data, and we're starting to mine back into a lot of the data taken during regeneration. So it's characterization data rather than really accurate phenotyping, but we're starting to move back into that, and there are a couple of other groups working in Mexico on highland maize adaptation, teosinte adaptation, so we should have that data available by the end of the year to report back.
Thank you, Sarah. 1 more question, burning question, Jean-Marcel.
Just shout, Jean-Marcel.
Okay, Jean-Marcel Ribaud, Generation Challenge Program. I mean, in maize, we are very lucky to have also quite a lot of historical material that can be used for changes in allelic frequency. And at CIMMYT, you have pools from the gene banks, population that have been improved, a lot of things. Maize is also well known in terms of migration, moving from different ecological zones. So is this also planned to low-hanging fruit, perhaps, to look at, what kind of alleles have been, selected, increase their frequency, looking at resequencing randomly some of those populations, and again, find the haplotypes that you want to select for in your elite germplasm?
Yeah, I mean, we're very lucky in that respect that a lot of the historic pools and populations that CIMMYT generated from the 60s onwards, every other cycle is a gene bank entry, so we have an accession for that. So we have included those in among every other accession that has been evaluated genotypically. We have characterization data. We're now digitizing a lot of the data generated under the LAMP project. So we're starting to merge those together to look for these broad selection sweeps. And many of the CMLs are derived from these same pools and populations, so we can look at the genetic drift or genetic selection as we've gone forward with these cycles and see really what has been fixed and pulled into the CMLs. So yeah, this is my fun job this year.
Great. Well, let's give our speaker a brief round of applause. And